Our study uncovers mechanisms occurring in neurons that contribute to Alzheimer's disease. Alzheimer's disease is the most common form of dementia, and it's the sixth leading cause of death worldwide. In the brain, this illness involves hallmark pathology, plaques and tangles, that impair neuronal function, as well as cause the eventual destruction of neurons. In our study, we asked, what is responsible for the transition of neurons from a healthy state to one with Alzheimer's disease? To determine what goes wrong in neurons of Alzheimer's patients, we looked at areas in the DNA called enhancers. These enhancers act like volume dials for gene functions. They're important for turning up or turning down the activity of genes. At the enhancers we looked at, the DNA layer is modifiable by an epigenetic code, and this epigenetic code is responsive to the environment and aging. When we looked at the epigenetic code at every single enhancer in the brain, we found places in the code that was abnormal in Alzheimer's patient neurons. In fact, there was a widespread overactivation of many enhancers, which impacted the function of genes. The genes we found affected in Alzheimer's disease neurons affected the pathology of disease, but we also saw pathways involved in the formation of new neurons. This was pretty surprising to us, since neurons are supposed to be done dividing. In the part of the brain we were looking at, there should be no new neurons forming. It turns out, when mature neurons turn on pathways that activate their division, that kills them. So our study identified genes that were responsible for the eventual death of neurons in patients. An enhancer area we found to be highly changed in Alzheimer's disease was in a gene called DISCOM1. We found that this enhancer regulates the neighboring gene, BACE1, which is responsible for the formation of plaques in the Alzheimer's brains. Abnormalities of this enhancer in DISCOM occurred very early in Alzheimer's patients, and moreover was linked to the severity of Alzheimer's plaques, tangle pathology, and the development of cognitive and memory deficits in patients. Many of the changes we saw in enhancers in Alzheimer's disease occurred very early on in the illness, before the pathology reached that part of the brain we were looking at. This suggests that these changes might be important in initiating or driving the disease process forward, but we're looking into that further as well. We also know that the epigenetic code is very sensitive to aging. In neurons, we found that the code was accumulating changes with age, but also these changes were accelerated in neurons of Alzheimer's disease patients. Epigenetic changes occurring with age might prime the neurons to progress to an Alzheimer's disease state, and if that activates genes that are responsible for cell division, that leads to the cell death that we see in Alzheimer's patients.